let's recap pyruvate metabolism in aerobic respiration. So in pyruvate metabolism, which is a 3-carbon molecule in aerobic respiration, pyruvate is converted to what? Acetyl-CoA, which is now a 2-carbon molecule by an enzyme known as pyruvate dehydrogenase complex. So pyruvate dehydrogenase complex converts pyruvate to acetyl-CoA. And remember, whenever we are trying to convert acetyl-CoA, whenever we are trying to convert pyruvate to acetyl-CoA, that simply means something, that we don't have much energy in our body. We don't have much ATP in our body. Therefore, since we don't have much ATP, therefore we have too much or high level of what? NAD positive and an ADP. When there is high level of ATP, there is low level of ADP, and when there is high level of NADH, there is a low level of NAD positive. So therefore, when our energy is low, NAD positive will start stimulating this reaction. ADP also stimulates this reaction, and decarboxylation occur from 3-carbon to what? 2-carbon to form acetyl-CoA. And NAD positive is converted to what? NADH, which will now go to the matrix of the mitochondria to undergo electron transport chain to produce energy. And recall that whenever high ATP level is in our body, that simply means we have too much acetyl-CoA. And whenever we have high level of acetyl-CoA, this pyruvate dehydrogenase complex is inhibited by ATP molecule. It is also inhibited by pyruvate dehydrogenase kinase by adding phosphate group. Don't forget those things. So we have treated this in our pyruvate metabolism. Go back there if you're not understanding what I'm saying. And this process is an irreversible reaction. The process by which we convert oxaloacetate to acetyl-CoA, it is an irreversible reaction. Take note of that. So let's start. Now we have formed our acetyl-CoA in aerobic respiration, by which we convert pyruvate to acetyl-CoA by pyruvate dehydrogenase complex. Now this pyruvate, which is 2-carbon molecule, will now come and react with oxaloacetate, which is 4-carbon molecule, and it is stimulated by an enzyme known as citrate synthase. Citrate synthase, the word synthase should give you something. Citrate synthase means it's help in formation of what? Citrate, by joining acetyl-CoA with oxaloacetate to form citrate. Therefore, citrate synthase means we are forming citrate. Now, whenever we have too much citrates, whenever there is high level of citrates in our body, these citrates will now start inhibiting citrate synthase. And when citrate starts inhibiting citrate synthase, it will now reduce the way by which the conversion of acetyl-CoA and oxaloacetate is converted to what? Citrate. And whenever we have this high citrate, ATP will also come and start inhibiting this what? Citrate synthase. So when we have too much or high level of citrates, ATP starts inhibiting citrate synthase, and citrate itself also starts inhibiting citrate synthase in order to prevent the formation, or in order to prevent the more accumulation of citrates. Now from there, another enzyme called aconitase. Aconitase will now start converting citrates to isocitrate. So whenever there is high level of isocitrate, this isocitrate will also come and return and start inhibiting this reaction. ATP molecule also starts inhibiting this reaction. Just have it in mind that citrate synthase converts citrate by sticking or joining acetyl-CoA together with what? Oxaloacetate. And aconitase now converts citrate to isocitrate. From there, from isocitrate, it is now converted to α-ketoglutarate by an enzyme known as what? Isocitrate dehydrogenase. So whenever you hear the word dehydrogenase, just know that we are converting NAD positive to NADH. So that is its implication. We are converting NAD positive to NADH. And isocitrate dehydrogenase converts isocitrate to what? Alpha-ketoglutarate, which is what? 5 carbon atom. So it have moved from 6 carbon atom to 5 carbon atom by decarboxylation. Decarboxylation is the process by which we lose carbon dioxide by which we lose carbon in form of carbon dioxide. So the carboxylation, we are losing carbon in form of carbon dioxide. Carboxylation, we are adding carbon in form of what? Carbon dioxide. And isocitrate dehydrogenase stimulates this process and also inhibiting depending. So whenever there is accumulation of acetyl-CoA, this acetyl-CoA starts inhibiting isocitrate, and isocitrate will now start inhibiting or start reducing the amount of alpha-ketoglutarate by what? Inhibiting the process by which we convert isocitrate to alpha-ketoglutarate. So high accumulation of alpha-ketoglutarate leads to inhibition by what? ATP molecule. This ATP molecule starts inhibiting alpha-ketoglutarate. This alpha-ketoglutarate starts inhibiting isocitrate dehydrogenase. Isocitrate dehydrogenase stop this formation of isocitrate to what? Alpha-ketoglutarate.